The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, this is episode 82 of the eLife Podcast. I'm Chris Smith from The Naked Scientist. And this month, a DNA technique to track when one population merged into another and what it's revealing about human history, evidence that squeezing through tumours makes spreading cancers more malign, and moths that change their taste for what smells nice after they've mated. But first, when people live together, inevitably they share things. And that also goes for the microbes, dubbed the microbiome, that live on them and in them. But what happens when people and animals share the same space? Do they trade bugs too? Well, that's what Cornell's Andy Moeller has been trying to find out.
One of the major ways we're impacting wildlife is through urbanization, the spread of cities over the landscape. And we set out to answer this question of whether urbanization and close contact with humans is actually leading to the spread of microbes from humans into wildlife.
Usually when this happens and there's a jump from wild animals into humans, the outcomes are not good. And one product of that is things like monkeypox, which is causing a headache around the world at the moment, and COVID possibly. So are we talking about pathological spread, us giving animals things that make them unwell, or are we talking about a more innocuous thing where I happen to coexist with an animal and some of the bugs that live on me and in me become incorporated into the microbiome of that animal too?
That is the key question, I think. Anytime a microbe from one host transmits into another host species, where these are two lineages, the microbe and the new host, that have never interacted before, many times you can have very negative effects on the new recipient host. We don't know in this case if the bacteria that we find shared between humans and cities and wildlife in cities are deleterious or harmful for the wildlife. But there are reasons to at least speculate that receiving a microbe from a distantly related host could end up being bad for the recipient.
How did you study it then?
What we did is we went out and sampled fecal samples. So we took feces from wildlife living in multiple different populations in multiple parts of the world, living either in cities or in rural areas, more natural areas. And we simply asked, do the wildlife living in cities share more microbes with humans than the wildlife living in rural areas? And we found over and over again this increased sharing between urban wildlife and humans compared to the levels of sharing that we observed between rural wildlife and humans. And so the inference from that was that wildlife seemed to have actually acquired some of these human microbes.
Is it the interaction and proximity to humans, or is it that, for instance, I eat a chicken and that does something to my microbiome, I chuck the chicken bone in the bin and along comes Mr. Fox. He raids my bin and eats the chicken bone, and the chicken has the same impact on his microbiome as it did on mine. Do we know whether that is the process, or is it that I leave some of my microbiome on my chicken, Mr. Fox comes along and he picks up my microbiome directly?
That's a great point. So we can't definitively distinguish between those two alternative explanations. So I should back up. I've sort of been favoring the transmission explanation, but it is possible that there's parallel selective pressures, for example, like you mentioned, due to diet or some other shared environmental factor that leads to the same bacteria being selected for in both wildlife and humans in cities. I think that explanation, although possible, is less likely. It's a little more complicated, less simple of an explanation than there's this sort of cloud of human-derived microbes that's floating around us everywhere we go, our trash and so on, our waste, and that the wildlife in cities are likely just picking up microbes from time to time from humans.
Is there any evidence that it might be impacting the health of those animals? Because researchers in Australia did a similar sort of study looking at seagulls in the last few years, and they were concerned to see that seagulls, probably playing around in sewage outfalls, were picking up antibiotic-resistant forms of microbes, human forms. But they weren't necessarily becoming unwell in the process. They were just acting as vectors, and they were spreading these things around. Do we have any insights, or what does our instinct tell us these acquisitions of human microbiome makeups might be doing to these animals?
Yes, so our study finds this very significant pattern of increased sharing of microbes in urban settings, but we don't investigate or have any data to speak to the health effects on the wildlife of having microbes that are also found in humans. It is important to note, however, that some of the microbes that we see in the urban wildlife that are missing from their rural counterparts belong to bacterial groups known to cause disease in humans. For example, very close relatives of Clostridium difficile, we see close relatives of that bug showing up in some of the wildlife.
So while there may not be direct or visible obvious health impacts on these recipient animals, it may almost end up with us parking some of our pathogens among that community, which may then in some way continue to evolve or amplify, but ultimately might find their way back into us again.
Exactly right. It is a possibility. If multiple species of mammal and other vertebrates are able to harbor these lineages of bacterial pathogens, you're completely right. It could actually help maintain genetic diversity in that pathogen and potentially allow it to evolve in ways that it wouldn't have been able to evolve if it didn't have those reservoirs in wildlife.
Has anyone done the equivalent experiment in humans, though? And you compare urban dwellers versus rural dwellers. Do you also see sort of more sharing of an urban microbiome in the people? And might that have an impact as well?
We do, actually. When we just look at the humans, for instance, living in urban settings, their microbiomes tend to be more self-similar than humans from rural populations. So we see kind of this parallelism and convergence in urban settings among humans and also among humans and wildlife species.
And turning it round then, how do we know that what we're calling a human microbiome associated with urban living has gone into an animal? Could it not be that the animals give it to the human city dwellers?
Right. So in that case, we can show that the microbes underlying this increased similarity between wildlife and human microbiomes in cities are also found in rural humans, but they're not found in rural wildlife. And so that observation tells us that these are more likely human-specific microbes or human-derived microbes than microbes that were already present in the wildlife before they moved into cities.
Fascinating, that, isn't it? Andy Moeller there. When we're trying to piece back the events of thousands of years ago that led up to giant leaps like the inception of farming or the colonisation of a whole continent, we rely heavily on the archaeology for clues. But thanks to a technique developed by Manjusa Chintalapati and her colleagues, genetics can also be brought to bear and enable us to see very accurately when, back in history, so-called admixture, the merging of one population into another, occurred. It can give us new insights into when postulated migrations, crucial to the evolution of the human race and practices like agriculture, did, or, as it turned out, did not happen.
When you have two populations which admix, then the offspring has one chromosome from each parent, and these chromosomes get shuffled when the offspring gives rise to the next generation. So basically the chromosomes fragments get shuffled every generation. So you can actually use recombination as like a molecular clock. Based on the length of the DNA fragments you inherit from each of the parent, you can ask when did the admixture occur? Because if the fragments are a bit longer, then admixture could have occurred, few generations ago. Or if the fragments are much shorter, then the admixture many generations ago.
Very elegant idea. So I presume, though, this is dependent on knowing some sequences which are exclusive to each of the populations, so you can tell what's been added, because if they're the same, you wouldn't have any kind of footsteps to follow, would you? So how do you know what is unique to each of the populations that have merged?
You do need information of both the parent populations, basically the genome you want to date admixture in, and also the information of the populations you think the admixed genome has information from. Let's say you have an example of African Americans, you do need information of source populations, which are the European populations and also like the African populations. And then you can ask when did these source populations admix.
And are you considering the entire genetic code when you do this, or do you just look for individual little bits and you follow those little bits as almost like markers?
We actually follow the entire genome. So basically we process the entire genome of an admixed individual and ask where are these blocks of DNA coming from, let's say European or African, and then we ask how long are these fragments. And just based on an estimate of how long these fragments are, we can actually go back and estimate, because this actually follows theoretically an exponential distribution. We can fit an exponential distribution to this segment length and then find out when that mixture actually occurred.
Does it not matter where these bits are on the chromosome in terms of how likely they are to get diluted and recombined and that kind of thing? Does your method take that into account?
Yes, that's a very good question. It matters because there are recombination hotspots and cold spots in every genome. So some of the fragments can get much shorter just because there could be more recombinations, meaning there's more DNA breakage there every generation. So we do use a recombination map. We actually have information where this recombination occurs, and we do account for that.
This is brilliant, but I'm frightened almost to ask, does it really work? And how do you know it works? Have we got a gold standard against which we can run this, so we can say, well, we know when this genuinely happened. Now we're going to ask, what does your genome tool return, and do the two agree?
Yeah, there are examples where you know some of the history is documented, right? So you can use them as positive controls. But a much more better way of evaluating your method is always simulations. We actually simulated admixture between European and African ancestries, let's say 100 generations. When we apply our method, we date back 99 generations with 3 standard error, let's say. And this is true when we change the proportion of mixture, when we change the time of mixture, when we change the sample size of the population. So we tested our method for several scenarios, and it works robustly in many scenarios.
It's almost like the genetic equivalent of carbon dating, isn't it? In the sense that, you have a half-life and the radioactivity is falling by a predictable amount each time. But it gets less accurate the farther back you go because the graph flattens out. Now, does your method fall prey to the same thing where you've got a sweet spot, but once you start to get to a long time, the noise becomes more pronounced than the actual signal? So what are those ranges?
Yeah, that's a very good question. You're reading my mind. When the fragments get very, very short, it's very hard to date back the time just because there isn't any information, right? The resolution of our method is actually up to 300 generations. So if you approximate the generation time to be around 28 years on average in humans, that is around 5,000 years. But the novelty in our method is that because it can work with many degraded data, we can apply to ancient sample, let's say, which lived 5,000 years ago, and the admixture in that sample could have occurred 5,000 years ago. So now we have ability to look at admixture events which occurred, let's say, 10,000 years ago. That's the exciting part of our method.
I mean, it hasn't escaped my notice that this is mapping on beautifully to the times when ancient peoples were doing really interesting things and moving around a lot, embracing new ways of living, farming, agriculture, and so on. Are you already probing some of those important questions around when people did things historically?
Yes, exactly. Just to give you a background, all present-day Europeans can be modeled as a mixture of 3 populations, which are the hunter-gatherers, which come from the Mesolithic time period, Anatolian farmers from Neolithic time period, and pastoralists from steppe, from Bronze Age. So we could actually date back the time of formation for farmers and steppe pastoralists using dates.
We have vague ideas based on a range of measures for when those different things and those different populations and different groups arose. Do our current predictions, based on those proxy measures, do they agree with your method? Have we got it right, or have you found with this that, in fact, there are some gaps?
We know that agriculture in Anatolia actually dates back to a time of around 8,000 BCE, right? There's been a debate in the field saying that the agriculture in Anatolia reached there probably because of the movement of Iranian farmers, which was originally where agriculture comes from. But using our method, we could actually date back the gene flow of Iranian farmers in Anatolia to around 10,000 BCE. That is way before what is documented, suggesting that probably it was just not the movement of the people themselves, but probably the techniques got diffused over time. So it could have been, probably the hunter-gatherers locally transitioned to agricultural subsistence. So using our method, we could actually give a precise timing of admixture between 2 groups, which could shed some light on how farming actually reached Anatolia.
Manjusa Chintalapati, she's at UC Berkeley. You're listening to the eLife podcast with me, Chris Smith. Still to come, how moths find the flowers and plants that they need among a mishmash of potentially misleading odours. But first, to a remarkable observation about the behaviour of cancer cells. When they spread or metastasise, inevitably they become much more robust and less vulnerable to chemotherapy or the assault of the immune system. Now, many had speculated that this is just a product of the cancer equivalent of survival of the fittest. But Gabriel Ichim, who's at the University of Lyon, has found that fighting their way out of the tightly pressured environment of a tumour and squeezing their way into the bloodstream does something to the DNA of these cells, endowing them with their more resilient profiles.
Cancer is very dangerous when it spreads through the body. And at this stage, it's basically incurable. These cancer cells, they are resistant to cell death, but we don't know why. The spread of cancer through the body is very stressful from the point of view of mechanics. For the cells to spread, they really have to squeeze through tight places. Sometimes they are so stressed, they will explode. So putting these 2 points together, we wonder whether when they embark in this voyage, this stressful condition, actually is responsible for this resistance to cell death.
You don't think it's just a survival of the fittest thing, that some cells, which are a bit more robust and resilient and resistant to cell death, are ones that are best equipped to handle the stress of breaking away from the parent tumour and going to a new part of the body?
That was actually a main question raised by the reviewers. And we did a series of experiments that is not a selection of a pre-existing population, as you said, that are resistant to death, but actually it's something that is induced by this mechanical stress.
So this is effectively what doesn't kill you makes you stronger. It really is true with these cancer cells that the physical pressures applied to them to get to a new part of the body changes their behaviour.
Exactly. There's another saying is, it is not the strongest or the most intelligent who will survive, but those who can best manage change. And so it's how you better adapt to the stress that will determine how fit you are to spread through the body.
So we'll come in a minute to actually what they're doing, but what is it that actually induces the change in the cells? Or how do they detect that they're being stressed? And how does that alter their behaviour to give them these additional abilities?
My opinion is that the nucleus is the limiting factor. Sometimes it explodes, so the DNA is getting released. Sometimes there are DNA damages, and this mechanical stress, therefore, can be also mutagenic. So when you stress the nucleus, this will be perpetrated to the next generation of cells. So it's, I think, in my opinion, starts with the stress on the nuclear level.
When we started talking, you pointed out that cells that do this metastasis appear to become much more resistant to dying. So therefore, whatever the process is that changes them, it changes them in that way. So how have you unpicked what is happening to the cells to make them more resistant? And how do you link that to this idea that it is the stress and perhaps doing things to the nucleus of the cell that makes them change their behaviour like that?
First of all, I'll have to tell you how we created these stressful conditions. We basically put lead weights on the cells. We created the compression that is found in tumors. We also use a membrane with very tiny pores. Diameter is 3 micron. To give you an order of idea, the diameter of a nucleus, it's around 10 microns. And we found that only the constriction leads to increased survival of cancer cells. We wonder, okay, maybe how the genetic information is translated is changed. And for this, we've done RNA sequencing. And these cells modified their message. They are telling us more about how they move, how they resist immunosurveillance.
Given that you've now disclosed this, can we use this in a therapeutic way? Are there ways to exploit this phenomenon? Perhaps the detectors or whatever the processes are that make the cells behave in this way, we can preempt that this is going to happen and block the ability of the cells to respond in this way so that we have a new way to block metastasis, but also to target cancers.
Yes, what our study was very descriptive. We simply described this mechanism. We left the door open for researchers that maybe they will take these findings further and find, as you said, better therapies to target this new mechanism that we described. So we kind of open a Pandora boxes of ideas. Since mechanical stress is bronchogenic, we can consider strategy to soften the tumors. There will be no mechanical stress, and like this, they will not have this survival. There are ways to soften, to make tumors less hard. And then a last point, these mechanically stressed cells become invisible to our own immune cells. So there may be ways to remove this invisibility cloth and enhance their killing by the immune cells, but this is something that we didn't yet investigate, and then hopefully there will be other researchers that will take the study further.
Well, let's hope so. An extremely interesting observation too. Gabrielle Ukim there. Confronted with a mixture of scents, some coming from plants you can feed from or lay eggs on, and others from plants that are just a distraction, how do insects like moths track down and tell apart the sources of the useful from unhelpful odours? Well, it turns out they can discriminate very precisely between them, and their sensitivity also changes depending upon whether they've mated or not. Sonja Bischgnaden is at the Max Planck Institute for Chemical Ecology in Jena, Germany.
I wanted to know how moths can, just by using their sense of smell, find important plants that, for example, provide nectar for feeding, and plants that are suitable as host plants for their offspring. This is especially interesting in the case of moths, as most moths are specialized on specific plants. And if these plants are rare in the habitat, this means they have to find these resources against a huge variety of odors that are emitted from other plants that are not relevant for the moth.
But moths do have an exquisitely sensitive sense of smell, don't they? I mean, I remember one friend of mine pointing out to me that moths can pick up the smell of another moth at less than parts per billion concentrations. So why should it be a challenge for them to find the plants they're after?
The challenge is that these plants that they want to find release a complex mixture of odors, but plants that are not important release also mixes of odors, and the components in these mixtures might be very similar, just differ in the ratio or the concentration. So they have to somehow differentiate or distinguish important plants from the unimportant ones. We know that they find the plant, but we don't know exactly how.
How did you then pursue it to work out how they were doing it?
So we went to the natural habitat of one of these moths, and there we were at a field station of the University of Tucson. And there is a lot known already about the moth species that live there and also about the plants. So that means we could go there and we were sure that this is the correct habitat and there are all the plants there that the moth needs. And then we did collect the odors that are released by the plants in the field. And we did this with the plants that are important for our moth, but also we sampled from other species that were around, trees or from grass, flowering plants in the vicinity of the important plants.
So you end up basically with a smell snapshot of what the environment that these moths are operating in is like. How do you then work out how they respond to those different odorants that are in those samples?
Yeah, we used these samples as stimuli in physiological experiments, and we wanted to know what the antenna, so the nose of the moth, can detect. And then in a second step, we wanted to know how these odor mixtures from each of the plants is processed in the brain of the moth.
How on earth do you do that?
For what the moth can detect, we have a GC, that's a gas chromatograph, that separates a complex mixture into single components. A stimulus is then going to the antenna, and each time the antenna can detect one of these stimuli, we see this as an electrical amplitude. So then we know this chemical is detected by the antenna.
And what about the response in the insect's brain? How does it then interpret that constellation of signals coming in?
We focused on the first processing center, the antennal lobe. This is equivalent to the human olfactory bulb. It consists of 60 or 70 subunits, and each of these is targeted by one specific receptor type. And as each of the subunits has a different range of odors that it detects, in the end, if our moth, for example, has 70 of these subunits, the moth can detect much more than 70 odors by this kind of combinatorial coding.
And do you see then that the insect has a unique brain response for the plants that are high value for the insect and lower for the plants that are just contributing to the sort of smellscape that's going on in the environment?
Yeah, we did another kind of experiment. We did also test virgin and mated females, as we thought looking for a host plant where the eggs can be laid and the larvae develop is more important for the mated female. And there we thought maybe there we find a difference. And what we saw in the virgin female, especially the flower orders, were very much represented. But plants that are host plants were activating only one of these subunits and very weakly.
So you've got quite an interesting set of findings there then, which is sort of multidimensional, isn't it? In the sense that, A, they can detect the plants that are high value, that are going to be good hosts, or that are going to be a good food source. But that depends on whether they are looking for a host plant on which to lay eggs or not. And so it sort of switches in response to their mated state as well.
Yes, in the mated female, we expected that the host plant, so the important plant for egg laying, will be represented in a increased way in somehow, maybe more subunits or higher activation. But this was not the case. It was exactly the same. But the activation of the background plants almost vanished. So the moth didn't smell these background plants anymore. Only the floral orders that are important for feeding and also the host plant for their own larvae. But at the moment, we don't know exactly how the mating influences the olfactory system. So we don't know the mechanism.
Sonja Bischknaden. Working in Africa provides neuroscientists with opportunities that are not available in other continents. Populations in this region exhibit the greatest genetic diversity, they live in ecosystems with diverse flora and fauna, and they face unique stresses to brain health, including child brain health and development, as UCT neuroscientist Kirsty McDonald writes. This, she says, is a unique chance to develop ways to benefit the continent and the field in general.
Neuroscience in Africa feels like it is an area of massive potential. The African region is one of the last remaining areas in the world where there is a young and relatively growing population. So what we are trying to communicate is the importance for us of optimizing institutions and scientists to be able to answer questions which are important for our region, but also to be able to contribute to the global questions in the area of neurological and mental health and neuroscience.
Those are the opportunities then. How do we
Yes.
capitalise on them? What's it going to take to realise the opportunities and make the most of them while this window is open?
One of the things which is really encouraging about our area is that people coming from resource-limited setting have a lot of resilience and resourcefulness with respect to how to use equipment, how to solve problems. You know, if you don't have a ready-made answer, you try and address it yourself. And so that mindset of resilience and resourcefulness is a huge opportunity to harness. A number of elements have been put forward by the community as things which we can use to build this community. One is a model of having hubs of expertise and systems of understanding the spread of expertise across the region. Ironically, COVID has facilitated this level of communication because of the more widespread use of virtual methods of communication. So, for example, having a database where you know where all the MRI machines are in the country or in the region. Where are they? Who has access to them? Are they available for research or just for clinical use? Is there a possibility of adding more resources so that one can expand the community of people using a particular methodology? The other area which is critical is mentorship for students coming through the system to develop critical thinking and scientific citizenship being able to communicate your science clearly, being able to develop independence in your thinking, giving people experience of not only good mentorship, but being able to mentor themselves while in a mentoring process. So there are a number of things which are easy to do. They don't necessarily cost money, but can really help move the field forward.
What's it going to take to implement them, though? So you've given me the opportunities. You've told me what your aspirations are and why they matter. Now, what about the practicalities? What's it going to take to join up Africa? It's a big continent, lots of countries there. This is a huge job that you've just outlined. Yeah, huge potential, huge job, though. What's needed?
So what's needed are champions. The Neuroscience Institute in Cape Town aims at leading in this area, at bringing not only different people in from across the continent to be able to facilitate this emphasis on regional growth rather than just local growth. But there are other virtual networks which are also driving this. So, for example, there's a network called Camera, led by a scientist in Uganda. It's driving to run workshops in MRI work and get people speaking to each other across the continent so that they can ask questions and utilize equipment that is available to be able to answer relevant questions to the region.
So this is almost like a call to arms on your part. It's a call to action. Community needed. We need to build the network. This is what we have in mind. Call us. Let's start a conversation.
It is absolutely. It's call us or expect our call.
Kirsty Donald, she's at the University of Cape Town. And there we must leave it for this month. You have been listening to the eLife podcast that's produced by The Naked Scientist. There are previous editions, references, and also the full text transcripts for these programmes, as well as details on how you can subscribe to this podcast, if you don't already, at nakedscientist.com/eLife. The Naked Scientists also publish a weekly science programme that covers the latest leading science stories, and you can find that at nakedscientist.com/podcast. I'm going to be back in another month or so with another look inside eLife. But until then, from me, Chris Smith, thanks for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.